Please come up here. Thank you.
Well, thank you very much for this kind invitation to present. And I hope in light of that last discussion about almonds, which was a little bit of a downer, that I can present some good news. And one of the things I wanted to share is that the research I'm going to talk about was actually funded in part by you guys, by a grant from the California State Beekeepers Association. And this was something that we got 2 years ago, and so now we're starting to see the results and fruition. Well, my talk is basically how can we activate the honeybee's natural immune system against a very common infection, Nosema. So the big picture is that honeybees face major disease challenges, and although antibiotics like fumagillin are quite effective against some pathogens, like Nosema, resistance may in fact be growing. This is the work of Huang et al., and their research suggests that Nosema ceranae is more resistant to fumagillin than Nosema apis, which was the formerly most common infection from Nosema. Now, there's also something that's rather worrying as well. We know from studies with livestock, particularly cattle, that resistance genes in the gut of these cattle have now transferred to humans because they're identical to the ones that you find in humans. There is also, based upon this analogy, tetracycline resistance genes that have evolved in American fowl brood, which is commonly treated with tetracycline, and these now are found in us. Could be a pure coincidence, but it is also possible that there is a transfer of these genes. So for a variety of reasons, it would be desirable to use fewer antibiotics and to use less antibiotics. So the question is, can we activate natural bee immunity? The outline of my talk today is going to be what is Nosema. Then I'll be looking at larval infection, the first experiment, which shows that larvae can actually be infected with Nosema. Then experiment 2, immune activation, and then the next steps. So this is the life cycle of Nosema. Basically, it is a kind of fungus that reproduces by spores. These spores, shown here, penetrate the gut cells of bees, and they reproduce in the midgut of the bees. At a certain phase, there are so many of these that the cell explodes, and they're released into the gut, and they are defecated by the bee. And it's thought to be a disease that's passed from fecal to oral transmission. So basically, the cells that are pooped out then can infect other bees that are trying to clean the colony or somehow coming into oral contact. So why is it important? Well, it turns out that in national surveys of managed bee colonies, Nosema is extremely common, and it's routine for major beekeeping operations to treat against Nosema. It causes gut tissue degeneration, as we just said. It's associated with collapsed colonies. There are synergistic interactions with pesticides, and it has negative effects on learning and homing. And most interestingly, it's been shown to reduce honeybee queen health, and queen health is a major concern that we now have. Now, heavy infections can cause immune suppression. I should say that Nosema is usually not fatal to bees. Colonies usually don't die because of Nosema, but in combination with other health stressors, particularly those that involve the immune system, you have a situation where you have a weaker colony. It turns out that exposure to Nosema ceranae does initially activate the immune system. So bees have an immune defense, and this is something called the toll pathway. This is a complicated biochemical cascade, but basically the bee's immune system is activated and produces products that help fight the invader. The toll pathway is commonly involved, and these names that you see here are names for the genes that are turned on that are part of the immune system of the bees. Now, it turns out that bees that are resistant, and there has been a strain of Nosema-resistant bees that have been developed, they continue to have activated immune systems. So they can handle it, they can attack the Nosema, defeat it, and they don't succumb. It's only the bees that are not able to do this that eventually have serious problems. Now, I wanted to talk, therefore, about insect immune systems. They aren't like us. They don't have antibodies to a specific pathogens. So for example, we have all been exposed to vaccines very likely. And these vaccines enable our immune system to recognize this bacteria. And in the future, when our body encounters it, that immune memory kicks in and we're able to fight it off. Well, insects don't have anything as specific, but they are susceptible to something called immune priming, in that you can activate the immune system, you can activate the toll system that I just talked about, and as long as that remains active, you actually have protection. It's a more general kind of protection. In other words, maybe you activate against one type of bacteria, and now the immune system is going to try to fight all kinds of bacteria. But if you think about it, that actually could have some advantages. So in red flower beetles, we know that larvae that are injected with heat-killed bacteria, essentially like a vaccine, they actually have protection within that generation, so that individual that got the vaccine-like treatment. The same thing with wax moth larvae. Now, the most fascinating area in this field is something called transgenerational immunity. So what if you were able to give the queen a vaccine that she would convey the protection to her eggs, to her offspring? So bumblebee queens given a heat-killed bacteria actually had protected offspring. And the same thing is true in the study by Lopez et al. In 2014. And they actually challenged the larvae by trying to deliberately infect them. These are the eggs of that queen. And they found that those larvae were actually protected. Most recently, there's been an interesting breakthrough. So vitellogenin, the egg protein that the queen produces, can actually pass on immune recognition of these bacteria. And it seems that the outer cell wall, the skeleton of the bacteria, is passed on through the vitellogenin, allowing immune activation in the offspring. Now, this has not yet been actually practically applied, but it's an interesting theoretical breakthrough. Now, notice what I've highlighted here. Everything is about heat-killed bacteria. In fact, to our knowledge, nothing has been done with fungal diseases, which, when you're beekeepers, you realize is actually an important issue. So can we use immune priming against fungal infection? So our first experiment is looking at larvae. You might also notice in the previous slide I said we treated the larvae of beetles, of Lepidoptera. And we wanted to know if the larvae of honeybees could actually be infected by Nosema. Now, Nosema can infect larvae of many different insects. There are different species that infect each of these different hosts. So we have beetles here. We have Lepidoptera, so moths and butterflies. And we even have bumblebees, whose larvae are known to be infected by Nosema. Now, saying that they can be infected is a different thing from saying it's a very important problem. But previous research suggested that honeybee larvae could not be infected at all. And it was largely based upon these two studies. One is showing in a different species, Nosema apis, does not infect honeybee larvae. And the second one that's more recent that looked at newly emerged adults and tried to look for evidence of these spores in the midgut. So there are classically two ways to check for infection. You can use something called PCR or qPCR to look for genetic material of the infection, or you could just count the spores in the midgut. Now, one issue is that honeybees possess hygienic behavior. So if you have sick or diseased larvae, the nurse bees and workers can come in and remove them. So if they were doing this, it would make it harder to detect the infection. In addition, Nosema is one of these things that kind of gradually creeps up. Typically, the highest infections, millions of spores, are found in the very oldest bees, the foragers, not in the youngest bees. So it makes it hard to detect in these younger bees. Well, we wanted to test the hypothesis that Nosema ceranae is not an exception in the insect world, that it can infect the larvae of its host. And so our experiment was to raise larvae in a lab, literally inside petri dishes, because then we can exclude the fact that nurse bees might be trying to remove the sick larvae, and we can control all the other conditions. We would then infect these larvae with spores. So what we would do is we grow them in these cages, we feed these bees a high dose of Nosema, we then purify them, and we confirm with genetic tests that we do have Nosema ceranae that we're giving to our bees. This is the grafting, and this is an amazing technique, I think, that was developed with honeybees that allows you to grow them in the lab. So here you see the 3-day-old larvae being removed and placed in these petri dishes. Here you can see the petri dish these well plates with these little circles of larvae inside. And this brief video shows you the process. Now, one problem is that in natural honeybee colonies, at this stage, nurse bees come in every day and they feed the larvae and they take care of them. Well, we don't have the nurse bees, so our solution is to use undergraduates. So we impress upon the undergraduates that if you don't come in, if you're late, your bees might die. And this is what it looks like. So day 2, when we're doing the grafting, at day 3, which you don't see here, we actually apply the treatment, and I'll go over that in a moment. They're developing, and then they're transferred, and then they emerge as adults, all in these petri dishes. So here's what it looks like. So here they're feeding the larvae, and they're removing the old food. This is something they have to do every day. They're ready to pupate now. They have to be cleaned. They're sort of covered with this sticky brood food that we've been feeding them. And this is a very delicate phase. And then they're transferred to new, larger pupation plates. Notice everyone's wearing gloves. We have to maintain sterile technique. They're ready to pupate. This is what they look like, pupating bee. And then they emerge as these cute, fuzzy, newly born bees. They're transferred into cages, and we let them live out their entire adult life. When they die, we freeze them, and then later on, oh, and by the way, this is the process. Here you can see the cages with the different treatments. In the back, these containers have very highly controlled humidity conditions because you need different humidity for the larvae and then for the adult phase. And then finally, we have undergraduates who also come in and do the rather laborious work of counting all these spores under a light microscope. So here's the first experiment that we did. Can Nosema actually infect larvae? So we have something called a negative control. We're giving the bees sterile water. They should not be infected with Nosema. We then have heat-killed spores, 40,000. This is called a spore retention control. And we did this because our initial paper was rejected, because they said, how do you know what you're counting in the bees is not just what you fed them? And so we thought, okay, well, we'll feed them dead spores. And if they do defecate before we finally look at them, but if it's possible that somehow these are not defecated out, then the heat-killed spores should actually show positive levels in the adults. Then we have our 2 treatments, 10,000 live spores and 40,000 live spores, which are actually low compared to what is typically used in the literature for these kinds of studies. We would look at the adult spore content and measure how long the adults would live. So these are the initial results. So you can see that about 14 days, so we have 0K is the control, 10K is the low dose, and 40K is the high dose. Mortality is higher in the higher doses that these were given. So there is an effect of adult longevity based upon the larval treatment. They weren't living as long. Now, why was this? Well, we also looked inside the gut. So we took bees, we dissected out the midgut, we embedded them in paraffin, and we sectioned them. And in these hundreds of sections, we carefully went through and we counted the number of cells that are infected. So here, these dark purple ovals are actually Nosema spores. And in a way, this is probably sufficient because you can see the spores inside the cells. They don't get inside the cells unless they're infecting the cells. And we found that 41% of our pre-pupae had these spores, and on average, in bees that were infected, 52% of their midgut cells had at least one spore. So here's the data on these spore counts. Now we looked at pre-pupae, so we dissected out their guts, and these are the spore numbers. Not huge numbers, and they are less than what we fed them, but that's what you would expect of Nosema. The infection grows with age. However, notice the autoclave control, these bees that were fed dead spores, they had no spores at all when we looked at them as pre-pupae. So what we fed the bees was defecated out. And what we're looking at here, even though it's low, is actually the result of an active infection. And of course, that's confirmed by our histology, looking at the microscope, seeing those spores inside the cells. Now, this was at adults, so they lived out their natural life, well, as natural as it can be inside a plastic box. And we then looked and saw how many spores they had. Again, the controls, no spores, or very negligible spore counts. The 10K dose has about 15,000 spores, so more than we actually fed them. But what's interesting, and I've highlighted this in red, is that the larger dose actually has a significantly lower level of infection. That's odd. We gave them more living spores. They should be more infected. But there's one possible explanation that we wanted to pursue, which is maybe we challenged their immune system so that at 40,000 spores, they actually responded and were able to protect themselves a little bit. So a natural extension of this is to say, okay, let's test this immune hypothesis and let's focus on using essentially a vaccine, the autoclaved control. So in summary, for this experiment, we showed that they can infect honeybee larvae, and the larger dose of 40,000 spores may have activated the larval immune system. And this takes us to the second experiment on immune activation. So this is the work of Matt Endler. I forgot to mention, although you saw his picture, that Darren Erie, my previous master's student, did the work on showing that Nosema can infect honeybee larvae. So Matt graduated last year, and this is his work. So here we have a somewhat more complicated treatment. I'm going to walk you through it. So this is what the larvae get in 3 days. They're getting sterile water, or they're getting our immune priming treatment. You can think vaccine. By the way, I'm kind of moving carefully around that word vaccine because it's thought not to be appropriate for use with insects. But it is, in many ways, when you think about what we're doing, very similar. Then we challenge them. As 1-day-old adults, we give them sterile water. This is called a negative control because they only got sterile water. They should not be infected. This one is a positive control because now when they're 1-day adults, they're given live spores. And other research has shown that this should lead to high levels of infection when they die as adults. We then have our immune priming given sterile water. These should not be infected. And then we have the most interesting treatment of all, immune priming plus 10,000 live spores. We're challenging their immune system saying, can you take it? We're going to give you the pathogen right now. And our measurements, again, are the same. We're looking at spore gut contents. We're looking at longevity of the adults. And our prediction is that the inactivated Nosema spores, basically our vaccine-like treatment, will activate the immune genes. Now, it's something I haven't presented yet, but I will show now, and provide protection. And remember, these immune genes are the ones I talked about earlier in the toll pathway. So, first, before proceeding, I should say that the adult longevity did not show any significant differences, which is good because it meant that our vaccine wasn't hurting the bees, but the bees that were actively infected should have lived less long than the bees that were the controls. And we think that part of it is that by artificially rearing the bees inside petri dishes, they don't live as well as adults. And we're working on that in our next experiment that I'll talk about. But the main interesting results are here. So these are different immune genes in the toll pathway. These are control genes. They're similar genes, but they are not involved in insect immunity. And what you can see is that when we give the immune priming treatment, the levels of these bars are significantly higher. And this shows that these immune genes were activated. And by the way, these immune genes have all, in other studies, been shown to correspond with the natural honeybee immunity against Nosema. Now, notice I'm saying fed no spores. These bees were never given any live spores, and when we dissected the same individuals, we found no spores inside their guts. So these genes are being turned on, not because they're infected with live spores, but because they were given our vaccine-like treatment. And finally, this is the most interesting result of all. This shows the number of spores per bee upon adult death. So here are the controls, bees that were never given live spores. They are showing actually 0 levels of spores as adults. The bees that were given sterile water as larvae and then 10,000 live spores as adults, they have about 250,000 spores per bee on average, which is pretty high. The bees that were given the immune priming treatment as larvae and then challenged with live spores have on average about 3,000 spores. So significantly fewer spores. And therefore it seems that the immune priming treatment was quite effective. So the conclusions for this experiment: there are no effects in adult longevity. It's something that we plan to work on. Immune genes, abeicin, increased 20 times, and defensin 1 by 4 times. And here's the key result. We reduced adult infection levels by giving them this immune priming treatment by 97%. And the infection rate, this means the percentage of bees that had even one spore that we could count, by 71%. So average infection levels 97%, infection rate by 71%. Okay, so what are the next steps? And this really relates to the practicality of this. One question is, can we immune prime larvae that are naturally reared in colonies? Because the in vitro technique, although it's highly controlled, isn't very natural. So we're following the same technique, and this is the work of Alex Niskovic. He's a current master's student. What we're doing is we're giving our vaccine-like treatment directly to 3-day-old larvae inside the comb. We then photograph and mark the comb, we stick it back in the colony, and then we come back when it's capped brood, we put it in the lab in the incubator, and when they're newly emerged as adults, we put them in the cages, and everything is done as previously. Except now the nurse bees are taking care of the larvae themselves. We don't think that hygienic behavior will be a problem because these larvae are being given dead Nosema. They're not actually being infected. But there are some other variabilities. There's other variables that could affect the result.